Hi everyone, welcome back to A View On, Lonza's podcast on the latest science and trends in pharmaceutical manufacturing. Today, we look at new frontier in respiratory medicine, spray-dried mRNA therapeutics that can be delivered directly to the lungs or nasal passages and stored at room temperature. We've all seen the power of mRNA demonstrated through COVID vaccines. But what if these fragile molecules could be stabilized, packaged for easy inhalation, and used to treat, not just prevent, respiratory disease? Our guest today, Carsten Rudolph, CEO and co-founder of Ethris, explains how their lead candidate aims to do just that by restoring a critical immune function that's often impaired in asthma patients. Their lead candidate delivers mRNA that locally boosts innate immunity in the lungs, a strategy that doesn't target a specific virus, but strengthens the body's first line of defense.
What we believe there could also be really great utility is in developing this as a stockpiling drug for being better prepared for the next pandemic.
From chronic asthma to future viral outbreaks, the science behind this approach could reshape how we think about mRNA as a therapeutic platform. This is A View On, a podcast brought to you by Lonza. Hi, Carsten. Thanks for joining the podcast. I'm really excited to talk to you today about a topic that covers quite a lot of what Lonza is doing as well. So we will be talking about mRNA, we will be talking about spray drying, about treating unmet medical needs. Before we jump there, I would like to ask you to introduce us to your company, your background, and your vision of developing mRNA-based therapies for your target indications.
At Ethris, we're a spin-out from the Munich universities, basically. I'm one of the founders of the company. I was heading a research team here in Munich at the Ludwig-Maximilians University in the Department of Pediatrics. And I was always focusing, of ideas of developing gene therapies for the treatment of genetic diseases. And we realized that maybe messenger RNA could be a very good alternative, basically, to use this for gene therapy treatment. And we did some work where we saw, indeed, messenger RNA is very efficient in delivering mRNA into the lungs. But we also saw that there are some limitations around it. And we all knew this now from the COVID times, that messenger RNA can be immunogenic because it mimics a virus, basically. So we did a lot of work around that, and we found a technology to circumvent this. And this was basically then the birth for Ethris.
What motivated you to start a company?
We thought, my co-founder and me, that if we don't found a company, then maybe our technology that we developed at academia would just end up in a drawer. And so we were brave enough to say, let's found a company. I'm very happy now because we have now just completed our first clinical trial and move into a phase 2 study. But that's the background of our Ethris as a company a bit more than 10 years ago.
That sounds fantastic. And it's exactly what I wanted to ask about. So you mentioned 10 years ago. That means that the idea for using mRNA for treating respiratory diseases came before the world was hit with COVID, right?
That's absolutely correct. So we were at the very beginning starting to look into this, with also we know all the companies now who develop these fantastic vaccines, but we always looked into developing therapeutics. With messenger RNA, but not so much in the vaccine space.
That's really exciting because I think many of our listeners may already be familiar with mRNA, but rather as the vaccine element. But today we will really be focusing on the use of mRNA as a therapy, which brings me back to Ethris. We mentioned that you are focusing on respiratory diseases, pulmonary diseases. You also mentioned that you started your work around pediatrics. Were you originally trying to develop gene therapy specific for children or pediatric populations, or when you founded Ethris, was the scope more expanded and wider?
No, it's exactly where we come from. And that's my background, what I mentioned before. I was working in a department of pediatrics. And of course, Cystic fibrosis is a very common disease there. So that's why we looked in the beginning into cystic fibrosis. We pivoted away a little bit from that. And we have also now a program in a disease that is a little bit more unknown, I would say. But the kids basically have very similar symptoms. In this disease, it's called primary ciliary dyskinesia. Yeah, that's where patients suffer from mutations in genes that drive the motility of our tiny hairs that cover our airways and are responsible for transporting the mucus out of lungs. Yeah, so to clear our lung from inhaled dust and pathogens, etc. So the symptoms are very similar, and we are indeed working on a program for the treatment for these kids, but we further expanded our ideas, and our lead program that has now completed a single ascending dose study in healthy participants and is now embarking on our phase 2 study in asthma patients. So the idea behind it is to find a new way to improve the host resistance in asthma patients. So indeed, we started to look into gene therapies and into rare genetic diseases with our technology.
That's exciting. Would it be a curative approach or a long-lasting therapeutic schedule that would need to be followed?
I think that's an excellent question. No, it's a chronic treatment, because the messenger RNA persists only for a short time in the body and is then degraded. So it's not a genome editing component which is included in this drug product, but it's designed as we assume something like a weekly or maybe a bi-weekly a treatment that needs to be taken by the patient chronically.
Still fantastic. And I assume it would be administered in form of an inhaler, right? So in the comfort of their own homes.
That's exactly the idea. And that's very important so that it's a very easy to use handheld device. That's exactly what we plan to develop for these patients.
Well, fingers crossed. That sounds fantastic. Just a second ago, Carsten, you also mentioned that it's unstable, mRNA, and then it gets degraded in the body. It also poses challenges, right, when it comes to formulating or even during manufacturing. So usually thinking about vaccines that we've all seen, mRNA products are normally frozen and stored at extremely low temperatures. However, with your approach that we've already seen through the collaboration with Lonza, you are aiming to generate a formulation that's room temperature stable. What's the thinking behind this and how can this be achieved?
So the thinking behind it is, as we already discussed, is that since we focus on developing drugs for the treatment of respiratory diseases, there is always this critical step involved where you have to nebulize these mRNA formulations so that they can be inhaled. And what we of course desire and want to have for the patients are then drug products that you can very easily use at room temperature, because patients are used to carry these easy-to-use sprays like they know from the asthma field or from the COPD field. And one prerequisite, of course, for this is that you have very stable formulations of your messenger RNA that you can store at room temperature. And there we have done a lot of work to achieve this. And I think we are on a very good way here, of course, also with Lonza, to further advance this technology to achieve spray-dried formulations that you can use to fill this in dry powder inhalers in the end for the patient to use the drug.
So one thing is to adjust the formulation to ensure the stability, right, of the mRNA. How about the delivery itself? So I assume the mRNA is encapsulated in a lipid nanoparticle, then sprayed, right? Do you add anything to the formulation that would help mRNA reach its final destination?
Indeed. So what we do is, and you mentioned this already, these are lipid nanoparticles. So we also developed our lipid nanoparticles, or LNPs, how you call them, but they are not, derived from these ionizable lipids that are used in the vaccines. But we have developed cationic lipidoids, lipid-like molecules. And they come with the advantage that they have a higher packaging capacity for the messenger RNA. So that's very important and make them perceived stable, more stable. But they come also with the benefit that wherever you deliver them, they are retained at the site of delivery.
Interesting point, Carsten. Could you clarify what that means?
For us, this means, if we deliver this to the respiratory tract, to the lungs, we can really maintain the full activity of the drug product at the site where it's needed in the lung tissue, because it does not biodistribute into the body, which is also a difference from these ionizable lipid-derived formulations, where we know from the vaccines, although locally injected into the muscle, they're not retained there, but they distribute to the body. So that's a very important key element of our technology from these cationic lipidoids. And what we did is we developed what we call a stabilizer. So it's an excipient that you can add to these LNP formulations, and which enable really stabilizing against any mechanical stress.
Mechanical stress, do you mean like shaking a vial?
Not only when you shake a vial, doctors love to flick against the syringe, and we know from the commercial vaccines, one should not do it because they tend to aggregate, But this is, of course, also a prerequisite for not only nebulizing a drug product for inhalation, but also, when we think about the spray drying process where you have to spray the formulation, and when we also think about the manufacturing process, where you have the fill and finish process where these LNPs are flowing through these little tiny tubings, and where shear stresses acts on these particles. So that's, I think, very important what we've developed here to really fully mechanically stabilize these LNPs. But we believe that's a precondition really for the next steps that are needed.
We've just heard how developing mRNA therapies for inhalation comes with unique formulation challenges. Ethris tackles this by designing lipid particles that stay where they're needed, in the lungs, and by adding stabilizers that protect the particles from the physical stress of manufacturing and delivery. Together, these innovations help make spray-dried, room temperature stable mRNA medicines possible. So now we've covered the drug product itself, spray drying, etc. Could we also talk about the mechanism of how the therapeutic actually works in a human body? So following the basic dogma of biology, right? mRNA is there encoding information for a protein to be then made in a cell once it reaches its target destination. I wonder, how many proteins have you already worked with? How many have been encoded and tested?
Oh, we've tested many, many different ones, basically. But the most important ones, of course, that are most advanced is our lead program, ETH37. And here we have a messenger RNA that encodes for a protein called interferon lambda. And interferon lambda is a very central innate immune modulator of our mucosal surfaces, which is capable of programming the epithelium into a very broad antiviral state. So that's the really molecule that can restore the host resistance that is dysregulated, for instance, in asthma patients. And that's exactly for what a purpose we developed this program, to develop a drug product for asthma patients to prevent exacerbation.
What are those?
Exacerbations are these typical asthma attacks that patients suffer from, and where we have this sudden worsening of the disease symptoms like tightness in the chest, breathlessness, coughing, wheezing. And in about 80% of the cases, this is induced just by a viral infection. This can be a very harmless virus infection, but it's all in a sudden induces an exaggerated inflammatory response leading to this attack. And this is all because of their impaired host resistance of the epithelium. And this is exactly what we intend to restore with interferon lambda delivery, because interferon lambda, as a central host innate immune modulator, is dysregulated in asthma patients. So that's the concept of ETH47.
Could you then say that you're locally enhancing immunity through your therapy in the lung?
That's exactly what we do. We restore the innate immunity and host resistance in the patients, so that they can control a viral infection again and to avoid then downstream exacerbations.
Amazing. And you mentioned that the target population here would be patients suffering from asthma. Could this theoretically be expanded to the whole population and used as a prevention from basically all other respiratory viruses?
Indeed, it has great potential. It's not only for asthma relevant, but there are also other severe lung diseases where viral infection can lead to a worsening of the disease, like we know this from COPD, but we also know this from IPF patients or even cystic fibrosis patients.
And what about the prevention aspect?
What we believe where there could also be really great utility is in developing this as a stockpiling drug for being better prepared for the next pandemic. Because as you say, it's virus agnostic. So it would be great to have this at hand during the beginning of the next pandemic, so that we can prevent, at least in the vulnerable patients or population in our countries, from an emerging virus, as a pandemic virus, and better a bridge, the time until we have tailored vaccines available again. I mean, because that's what we learned from the last pandemic. You need to have something available very fast and quick, so to really contain the virus as possible. So, no, you say it as a great utility, this drug candidate.
Fantastic. Oh, I'm excited and I feel safer already. We have not covered yet the administration route, right, when it comes to your product. So we keep talking about pulmonary delivery, etc. But I wonder, if you talk about an asthma inhaler, this is done through the mouth. I'm myself an asthma sufferer, at least when I was a kid, I used to. However, I believe that you are targeting also nasal administration, right? So would that be then a nose spray or a nose inhaler?
That's a very good question you raise here. So it is a nose spray, because what we need to cover at the first instance is the entry site of the virus in our body, and that's the nasal cavity. Where the infection at the first place happens, and where then this exaggerated immune stimulation takes part and leads to the downstream cascade of an exacerbation. So indeed, but of course, for our rare genetic diseases, like I mentioned before, PCD, there we need to deliver this then with an inhaler device to the lung tissue. So it depends a little bit on the disease, whether we target nasal administration or inhalation.
Could you now take a step back and talk about your platform itself? Could you tell us a little bit more about the background of and I'm not sure if I'm pronouncing it correctly, is it SNIM RNA or do you say SNIM RNA platform?
Yeah, we usually say SNIM RNA, and it's an acronym standing for stabilized non-immunogenic messenger RNA. What we found is that a standard messenger RNA is quite immunogenic, which makes sense, because it could mimic a virus, and we have a lot of sensors of our innate immune system in our epithelia that detect this messenger RNA and then stimulate an innate immune response. And what we did here is we modify messenger RNA. So we introduce modified nucleotides into the messenger RNA to minimize the potential interaction with these sensors of the innate immune system. So that's a very important element when we speak about repeated chronic delivery to patients. We replace two standard building blocks, nucleotides, by modified nucleotides, but only to a very low degree. So you may just consider this a kind of light touch modification, but that's fully sufficient, to change the pattern of the RNA so that's not recognized by the immune system anymore.
And where would you see this SNIM RNA platform in the next 5 to 10 years?
Yeah, so of course, we are very optimistic. And in 5 years from now, we want to have basically already concluded a phase 3 study with ETH47, our asthma program. And we hope that we are then even already thinking about filing a marketing authorization. Yeah, but we also, of course, want to have the proof of concept for our rare disease program and primary ciliary dyskinesia. And hopefully also we want to have advanced our technology platform in the mucosal vaccine space, where we think of developing a nasally administered messenger RNA vaccine, which can come with the benefit of achieving, we call it mucosal steroid immunity. That means that we can also hopefully block transmission in vaccinees with that technology. So we have a lot on our plate, what we want to have achieved in 5 years from now.
Quite a busy schedule, I can tell, yes. One additional follow-up question on the vaccine spray that you are now talking about. Would you be targeting the standard respiratory diseases such as cold, flu, and COVID, or also go beyond that?
So there's a lot of room where we can use this technology. Right now, we mostly look into influenza to develop this. Also in a program that we now got funded from CEPI, where we have a target with influenza that we are working on.
Thank you so much for sharing your insights with us. I cannot wait to see where you are 5 years from now.
Thank you very much for having me.
Today, we explored how Ethris is advancing room temperature stable mRNA therapies designed for inhalation or nasal delivery, offering a new approach to treating respiratory diseases. These spray-dried formulations, developed in collaboration with Lonza, could also play a crucial role in pandemic preparedness by providing fast, virus-agnostic protection before tailored vaccines are available. Thank you for listening to a view on spray-drying mRNA-based therapies with our guest, Carsten Rudolph. Join us next time as we discuss emerging trends in the early development of pharmaceuticals. If you cannot wait, head over to lonza.com/a-view-on to listen to our previous episodes, subscribe to never miss an episode, and access additional materials and interviewee info. Bye for now.